So our next question is sketch and explain the structure of the DNA. So we already know DNA is a polymer, which means it's a long chain compound and it is made up of two strands, right? So it is double stranded is what we call it, and these two strands intertwined around each other, crisscross around each other to form a double helix structure, something like this. Now the two strands are mainly made up of alternating units of sugar and phosphate that forms its backbone. Now both the strand will not just be present around each other, they need to be connected with each other, and that connection is done by nitrogen bases, right? So we fill in the nitrogen bases over here and finally we label it. But before we go on to the labeling of the diagram, we need to understand that these nitrogen bases are of four types. We have the first one over here, cytosine, then we have the next one, guanine, then adenine, and obviously last one is thymine, right? So we write them as ATGC. Now we have to write the full form of the DNA. Obviously we know it is deoxyribose nucleic acid, and we have to also label the nitrogen bases and the sugar and phosphate backbone, right? So that's the base pair over there, and over here there is the helix of sugar and phosphate, right? So that is how you draw the diagram. Now how do we explain it? So first of all, we already know that our DNA is made up of nucleotides, right? So it is made up of a lot of nucleotides put together. So we know the name of the molecules, but in turn these nucleotides are made up of three smaller molecules, which we just discussed as sugar, phosphate and nitrogen bases. This is what we mentioned in the first point over here. So DNA is made up of nucleotide molecules, a phosphate group, a sugar group, and nitrogen bases are present in each nucleotide. Now that we have mentioned what the nucleotide is made up of, we will mention that the nitrogen bases specifically are of four types, as we already saw, right? So the nitrogen bases is of four types. What are they? Cytosine, guanine, thymine and adenine. Again, something that we have already seen before, right? Then we mention the order in which these nucleotides are arranged. That decides our genetic code. Okay, that determines our genetic code, and in the further points we will see that this genetic code needs to code for something. It codes for proteins which control our biological characters, right? In the next point we mention that most of the genetic code, most of the sequences in humans are the same, and humans have approximately 3 billion of these nucleotides, which is a lot of information for a human being, right? So human DNA has around 3 billion bases, and most of these bases are the same in all human beings. So more than 99% of the human DNA matches. Whatever difference we have, whatever variation we have is in that 1% difference in the DNA that we have. So can you imagine how much similar we are as compared to each other? Right? Anyways, so in the next point what we mention is the order of the nitrogen bases for the genes which instruct the cell how to prepare proteins. As we already discussed, the genes are going to instruct the cell which protein to prepare, how to prepare it, what is going to be the function of the protein. So all this information about what the cell needs to do is inside. Then in the next point we'll talk a little bit about the double helical structure that we've already seen in the diagram, right? So nucleotides are joined together to form two strands that twist to create a structure called as a double helix, right? So there are two strands. So DNA is double stranded, as we already know. They intertwine around each other and they form a twisted ladder like structure called as the double helical structure. In the next point we mention which nitrogen base attaches with which other nitrogen base, right? So before we do that, let's have a look at the word over here, nucleotides. We can split it into two parts, nucleo and tides, so that you do not make any spelling mistake over here. Alright, now moving on to the next point, which nitrogen base attaches with which other nitrogen base. So we already know adenine pairs with thymine and guanine pairs with cytosine, right? And in the last point, we mention that DNA is coiled tightly to form chromosomes. Normally it will be present in the form of chromatin fiber. So DNA binds with a few proteins called as histone proteins, and they are present in the form of chromatin fiber normally in the cell. But when the cell is going to divide, these chromatin fibers start condensing and they become tightly wound structures called as chromosomes, which are found inside the nucleus. So that is how you draw the diagram of the DNA, and this is the explanation of the points.